Hello everyone. Today we are going to talk about imaging brain activity with multiphoton excitation. We will cover the chapter 5 from 5.1 to 5.4 of the review paper. At the left side, we can see HeLa cells that are imaged using multiphoton sequential scanning. Let's move to the next slide. To explain a little bit about multiphoton excitation, we will cover two photon excitation. So the challenge in one photon imaging is that we cannot capture fast signaling events, and the brain tissue being highly scattering is not helping to image in deeper depths while retaining high resolution. In speed, we need high laser power. In two photon imaging system, the beam interacts with sample only in the focus region. At the right hand side, we can see some examples of multiphoton microscopy in blood flow and calcium imaging. So multiphoton imaging enables us to get an imaging depth of several hundred micrometers to few millimeters. Here we introduce a technique called multi-region two-photon imaging. Conventional two-photon imaging system have small field of views up to 1 millimeter square. That's not sufficient for capturing neuronal circuitry dynamics. So we can choose specific subregions within the large field of view and fast jump between them to maintain sub-second temporal resolution. Here is the diesel two-photon microscope that can measure a 5 into 5 millimeter field of view with subcellular resolution containing on the order of a million neurons. Let's move to next slide. Conventional two-photon microscope will focus the beam into a point called point source function PSF, but scanning a large field of view with this technique will take time, usually milliseconds. To increase imaging speed with different PSF is sculpted with to increase imaging speed. The PSF is sculpted with different beam modalities. For example, Bessel beams, Gaussian beams, enabling different sampling methods, line scanning, for example. Next, Fatemeh will continue.
Yes, so the system here that we're going to present is two-photon imaging in 3D with an acousto-optic lens. So here we first pre-select some regions of interest, and then with the help of an acousto-optic lens, we will guide the laser into the pre-selected areas. So the acousto-optic lens is composed of two pairs of acousto-optic deflectors. So with changing the wavelength of the sound waves in the crystal, the beams here will have different diffraction angles and will focus into a desired point. So all of this happens in 10 to 25 microseconds, and it can also provide an imaging volume of 400 by 400 by 400 micrometer. So this is the example we have here. But this selective imaging enables us to have high temporal resolution. So this is the example. Here we have 72 regions of interest, and with putting them together into one image, we will have simultaneous imaging of neural population, dendrites, spines, and axons. So it means that this technique is good for mesoscopic and volumetric two-photon microscopy, as we can also see here. So the next method is dense volumetric imaging with light beads microscopy, or LBM. So here an ultra fast pulse here is copied into 30 times, and each time with different energy, and each time with a delay from the other copy. It will penetrate into deep areas depending on the energy, and multiplexing them, each column will form a bead, right? So This kind of method will provide us volumetric recording. So here in this example there are 800,000 neurons, which we can see together, and that's a large neuronal population, and it can be used for neurocomputation. So to summarize, we talked about multi-region two-photon imaging, which gave us big field of views, and then we talked about sampling strategies for fast two-photon imaging that gave us fast imaging, right? And then we talked about acousto-optics, that because of the selective imaging, we had high temporal resolution. And then we talked about light bead microscopy, that could give us a volumetric recording, a dense volumetric recording. Thank you for your attention, everyone. Bye.